AP Biology, Quarter 1 Review, Ecology and Chemistry of Life. Review of science as a process. Remember that we learn things about nature using inductive and deductive reasoning. Deductive reasoning is taking what is already known and applying it to new situations. And we're using a systematic approach to answering questions about nature using empirical or testable evidence. The steps in the scientific method: make an observation, see something interesting about nature, ask a question about it, investigate some background knowledge, find out what is already known about what you're observing, develop a hypothesis. Remember, a hypothesis has to have three characteristics. It has to be in the form of a statement, it has to include the independent and dependent variable, and it has to be testable. If it doesn't meet one of those three requirements for a hypothesis, it's not considered a scientific hypothesis. So once again, it has to be testable in the form of a statement, not a question, and it should include the independent and dependent variable. Also, then after that, we can set up a controlled experiment where we only change one variable, called the independent variable, so that our results are only due to that one factor being changed. Then we can draw a conclusion, graph it to try to identify trends, and ask further questions if necessary. Remember, we have three variables. Independent variable is the variable the researcher changes to test a hypothesis. If we're doing a plant experiment and trying to figure out what wavelength of light works best for plant growth, then what we're changing is the type of light, and that would be the independent variable. Now, as a result of changing the type of light, we might have a change in the growth of the height. Or another way of saying that is the growth of the plant depends on the type of light we give it. So the dependent variable in that experiment would be the growth of the plant, or how high it grew. And then some control variables are things that we want to keep the same to make sure that our results are only as a result of changing the independent variable. Control variables might be like the temperature the plants are in, how much water they're getting, things that might affect the results. Who waters the plant really doesn't matter, because it wouldn't change the results as long as they're getting the same amount of water. The experimental and control groups are set up when designing a controlled experiment. The control groups are the groups or the conditions that are typical and we already know the results for. Typically, we're talking about a comparison group. The experimental group usually has the independent variable changed to the point where we don't know what the results are going to be. So, for example, with the plant experiment, if we gave it different colors of light, let's say red, green, blue light, and that's our independent variable. Dependent variable would be the height of the plant. Control variables, again, temperature, things like that. The experimental groups would be the green, blue, and the colors of light. However, we want to compare that to what plants normally get, just as kind of a baseline or a comparison of our results. So the control group in that experiment might be a plant with just white light to find out what normally happens with those plants. We typically use a line graph for graphing data over a period of time or a continuous set of data that's collected continuously. Typically, it's using time, however. Now, remember, we're talking time in increments, like 1, 2, 3 minutes, or March, April, May. We're not talking about just after 10 minutes collecting data. That would still make a bar graph because you don't have multiple points on your line. The bar graphs are used usually for quantities and comparison of those quantities. Validating results. We have three ways we validate our results, and you should know these. To make sure we have good results, we should have a large sample size, so we should use lots of plants. We should do multiple trials and get the same results each time. And again, with the plant experiment, we'd have to do it over and over and over again. And then we should publish our results and have it peer-reviewed by other scientists or other people that are interested in the work. This way we know that our results are valid. If you do an experiment and you get really great results, but no one can repeat those results, and there's some problems with your work, then no one else is going to accept those results. Other people have to be able to independently get those same results you did. Otherwise, it's not science. The conclusion, what was the evidence, and does it support or refute your hypothesis? That's kind of the essence of any conclusion. You also can discuss things like sources of error, things that could have been a problem, and any further questions. So let's go through this. Let's pause at this time and practice the experimental method. The independent variable in this experiment is the variable that was changed by the researchers, which would be the cell phone use. The dependent variable is what happens as a result of using cell phones, which is more tumors. So whether you get a tumor depends on whether or not you use cell phones. An experimental group would be the people that are getting the cell phones, so we're kind of wondering what's going to happen as a result of using those cell phones. And a comparison group or a control group might be people that never use cell phones, people like the Amish. All right, here we have an example of line graphs. Typically we use it for time. It can be used for other things too, like pH, but time is the most likely one. That goes on the x-axis if it's a line graph for time. Bar graphs are usually used for quantities. All right, so we have one of the more famous experiments back in time. We have the Reddy experiment disproving spontaneous generation. Back in the day, they used to think things like rats came from bags of grain, and that turns out to be not true. But how do we know it's not true? Well, also at the time, people used to think that meat produced maggots, and that was questioned by Reddy. So he had two groups. He had uncovered jars and covered jars, and then the flies were allowed to land on the meat with the uncovered jars. The covered jars flies couldn't get to. And if the meat produced the maggots, then in both cases we should see maggots forming on the meat. However, the covered jars had no maggots forming on the meat. So that disproved the idea that meat produces maggots. It should have produced maggots if that was the case, if meat actually made maggots. So what was the independent variable? The independent variable was the covering on the jars, or the thing the researcher changed to test a hypothesis. The dependent variable was the amount of maggots found on the meat. Or another way of saying that is the number of maggots depended on the covering on the jars. And then we had some control variables like the temperature of the jars, the amount of flies, the type of meat, the location in the room as far as light, no light. All those kind of things needed to be controlled in order to make sure our results are only due to one factor. You should be able to identify the independent, dependent, and control variables, as well as experimental and control groups, and draw a conclusion based on the information given on the AP test. Scientific hypotheses, theories, and laws. A hypothesis is kind of an educated guess, may not have any or much testing done. For example, life exists on planets with water. That's a hypothesis. We'd have to test that by actually going to those planets and finding out if life exists there. Theory, well-tested, peer-reviewed, broad explanation of nature that is used to predict events in nature. Now, theories are usually derived inductively. So inductive means taking specific observations in nature and tying them together to form a general conclusion. And typically you do experiments with that to test whether or not those explanations are valid. For example, every time we look at new organisms, we find that they're made of cells, and this led to the cell theory. If the squid is made of cells, the whale is made of cells, the trees are made of cells, the fungus is made of cells, bacteria made of cells, you start to realize that there's a general trend here. And as long as that holds up, as long as we still find things made of cells, the cell theory stands, and it's useful for predicting new things and the composition of new life as we find it. The theory of evolution predicts that living things change as a result of the selective pressures in their environment. So we can test that as well. We had the guppy experiment where we put the guppies, or read an experiment about how the guppies are put in different background environments, and as a result of predation, were selected for blending into the background. When there was no predation, then things like sexual selection took over, and having standing out spots actually was a reproductive advantage. And that's why it's useful. It can be used to make a prediction in nature. You didn't necessarily know for sure that the guppies would blend into the background if predators are present, but you could make that prediction because we have the theory of evolution. And then a law is a well-tested, peer-reviewed description of an event in nature that happens the same way every time. More narrow in focus than a theory. So theories don't necessarily become laws, and laws don't come from theories. They both have different purposes. For example, Newton's law of motion, second law is force equals mass times acceleration. It's a very narrow definition of what happens as a result of those three variables, force, mass, and acceleration. All right, characteristics of living things. This is arrived at inductively, taking specific observations in nature, forming general conclusions, trying to figure out what life's all about. Now, we have a mnemonic trick for this called Krugersk, cells, C-R-U-G-E for energy, R, respond, S, and C. So let's go through this. Living things are made of cells. That's the basic unit of structure and function in any living thing. Living things reproduce, make copies of themselves, even if they're a single-celled bacteria. Living things have the universal genetic code of DNA. Living things grow and develop, even bacteria. Living things obtain and use materials for building up the stuff inside of them, and they use energy as well. This is one reason why we don't consider viruses alive. They don't obtain and use their own materials and energy. They have the cell do all the work for them. Living things respond to their environment. Here we have leaves and stems of plants growing toward the light in a process called phototropism. Remember, that's a response to auxin on the dark side of the stem of a plant, growing a little bit longer and bending the plant toward the light. Living things maintain a stable internal environment. For example, we maintain temperature homeostasis inside, prevent from getting too hot or too cold. Things that are cold-blooded usually use their external environment or their behaviors to try to change the temperature of their body. And taken as a group, living things change over time. Now, we're not talking one generation, we're talking about several generations. And this is the explanation of why plants and other animals, any living thing, has the traits that help them survive in their environment. Plants that live in the desert survive because they have become adapted to the conditions of the desert. If you didn't have those survival adaptations as a result of mutations, then you wouldn't exist in the desert. That's why we don't find too many polar bears on the equator. So how do we study cells? The thing that you have to know about this, light microscopes are used for living cells, electron microscopes only for dead cells. Light microscopes can't see as small as electron microscopes, and that's something that you have to know too. Light microscopes work by light passing through the specimen. Electron microscopes work by coating the specimen with heavy metals like gold and then having the electrons bounce off of that and creating an image. So here we see the light microscopes can see down to some of the bacteria, smaller bacteria, as well as some of the bigger organelles. But electron microscopes can see down to the size of molecules, so they're very powerful. Now, the two types of electron microscope are transmission and scanning. Scanning gives you a 3D image. The transmission electron microscope only gives you a two-dimensional image. However, the transmission electron microscope can see smaller than the scanning electron microscope. The scanning electron microscope gives you some pretty interesting pictures, however. They give you those three-dimensional scary pictures of things like spiders and such. Here are some keywords that are important for behavioral ecology. Go ahead and review some of these. If you need to look them up in the back of the book, go ahead and do so. Also remember the Campbell's activity disc has the vocabulary list right there. You can just click on those words and get the definition instantly if you don't want to look them up. We started chapter 51 with talking about proximate versus ultimate causes. Proximate causes are whatever triggers a behavior. For example, the mating behaviors in these cranes will trigger the act of reproduction. The ultimate cause is why this is happening. And that's the evolutionary significance. So when you're asking yourself the ultimate cause for something, always ask yourself how this affects survival and reproduction. For example, with these cranes, if they have the right mating dance that triggers something in the brain that recognizes that as a mating behavior, the reason why that would be selected for is that's the thing that gets you mates. And as a result of the mating dance, you reproduce, passing out the genes for that instinct, and that is passed on to the children, and that will be in turn causing or resulting in more offspring that will also have those mating dances. If you don't have those dances, then you don't get mates, and you don't pass on those genes. All right, there's two types of behaviors. We have innate or instincts, and then we have learned behaviors that are modified by the experience of the individual during the lifetime. Now, both are triggered by a stimulus, and the stimulus is whatever is going to cause behavior. For innate behaviors, the way we know they're innate is they happen the same way no matter what the environment is. For example, if we have a moth exposed to light, the moth always is drawn to the light, and it will never change its behavior, even if it's something dangerous like a fire. Learned behaviors are modified by experience. So if you touch a fire, you're not going to touch it again, and a lot of animals learn also to avoid fire because it's hot, and they can learn from that experience. We talked about learned behaviors also for like birds and mammals, especially animals with a more developed brain. They can learn a lot better than an animal with a small brain, like an insect. All right, this is one of the innate behaviors, instincts, called a fixed action pattern, kind of a complex innate behavior. There's something called a sign stimulus, or something that triggers this series of instincts that will be unchangeable once they're started. For example, we have the red bellies of the male fish, and when another male fish with a red belly comes into view, no matter what, that stickleback as an innate behavior, will initiate the FAP of attacking it. And it's just an instinct to attack anything with a red belly. And there's even been those experiments with the painted wood blocks that these sticklebacks can't figure out are just painted wood blocks, so they attack them. The sign stimulus in this case would be the red belly. Now keep in mind when you're trying to figure out whether it's an innate behavior or learned behavior, especially simple animals, insects, most invertebrates really, other than maybe octopi, and even simple chordates like fish, will typically operate mainly on instinct or innate behaviors. When we get into things like birds and mammals, there's a lot of learned behaviors, still some instincts, but there's more likelihood of having learned behaviors because they are capable of learning with a more complicated brain. So insects, simple animals, almost all instinct. Directed movements, kinesis, both are in response to a stimulus. So whether it's kinesis or taxis, there's some kind of stimulus that results in that behavior. Kinesis is a simple change in activity or turning rate in response to a stimulus. So if you can imagine like a fly in a room, it senses some food in that room, it'll just kind of fly randomly until it kind of gets closer to it and is able to detect it a little bit better with its chemoreceptors. Here we have an example of a pill bug kind of randomly moving toward the dry or the wet environment underneath a leaf, which is also going to be away from the light as well. In this case, it's going to be the moisture causing the kinesis that will be the simple change in movement of that bug until it reaches the moisture. Taxis is a little bit more directed. It's also automatic. However, it's directed motion toward a stimulus or away from a stimulus. For example, we have fish. Remember, counter-current exchange is where the rate of diffusion is maximized as the water with oxygen in it passes over the capillary beds, which move in the opposite direction, which results in a concentration gradient at every step of the way, which gives you more oxygen out of the water. Now, if it's toward something, we call that positive taxis. And if it's away from something, it's called negative taxis. So the fish facing the water, so you get more water of the gills, is an example of positive taxis. An example of negative taxis might be a fire that animals have an instinct to run from. So that would be negative taxis, away from a stimulus. We can even add other words to this as well. For example, if a white blood cell detects the histamine released by a basophil and starts moving toward that area of infection, we could call that positive chemotaxis, or directed movement as a automatic result of the white blood cell recognizing those chemicals. Now, migration is still under genetic control, so this is still an instinct. It's an instinct to migrate. If the animals that typically migrate don't migrate, they tend to act very different and agitated. Imprinting is the one that kind of bridges the two gaps between, or bridges the gap between learned and innate behavior. This is an instinct to learn. So imprinting is where you have an instinct to learn on the perceived parents of the young animal, and it only really happens at a very young developmental age. So there's usually only a short window that happens in the early age of an animal that results in imprinting or learning from who they perceive to be the parent. However, it's an instinct to learn as well. So imprinting, instinct to learn. Example, geese following their mother or the person they perceive to be their mother to where food might be. And you can kind of think about this. The ultimate reason would be the survival advantage of following an adult that knows where the food is and getting food yourself if you're just starting out your life as a young goose. And then we have learned behavior. So we had innate behaviors like FAPs and taxis, kinesis. We had the imprinting, which was a combination of the two. And learned behavior, we have a couple different types. The first type is associative learning, or associating one feature of the environment, which is a stimulus, with another. Now, the first one we talked about was classical conditioning, and then we had a stimulus, which resulted in a reward or punishment. The classic example was Pavlov's dogs. What happened was the dogs were given a treat without a bell, and they started to salivate when they saw the treat. Then they were given just a bell, and they didn't salivate. After they were given the treat with the bell over and over and over again, they salivated, but they salivated because of the treat, not the bell, but they associated the bell with the food. Then, because they learned to associate one feature of the environment, the bell, with another, the food, they started to salivate just when they saw or heard the bell, even without seeing the food or smelling the food. And this would be an example of classical conditioning. Now, classical conditioning doesn't really require the animal to do much work to figure out something. They're just associating one factor of the feature of the environment with another. When Pavlov's dogs were given the meat with the bell, they didn't have to try different things to figure out if the bell would give them meat. It was just always bell, meat, okay, bell, time to salivate. In operant conditioning, it's more trial and error, so the animal has to work a little harder to figure things out. A classic example of operant conditioning is the Skinner box. Rats were put in a box with different paddles to push on, and they had to figure out on their own which paddles would give them food and which paddles would shock them. Since it was trial and error, they had to try different things. There was still a stimulus. When you hit the wrong paddle, you got shocked, and then you avoided it. If you hit the food paddle, you hit it more, get more food. However, because they had to figure it out, it's called operant conditioning. But there's still a reward or punishment involved. It's just that the animal has to do some work to kind of figure out which one works and which one doesn't. An example of operant conditioning would be birds eating monarch butterflies. When a bird eats a monarch butterfly, they learn that the taste is bad, it's poisonous, and they'll avoid any future monarch butterflies, and that would be a trial and error learning. They had to try something. It wouldn't be classical conditioning. Habituation, loss of response to stimulus. Basically what we're talking about is a type of classical conditioning. Over and over again, if you see a car as a deer, and at first you might run away from that car, but over and over and over again, those deer start to not be afraid of the cars. They just kind of stay off to the side of the road. And that's a result of being habituated or learning not to respond to repeated occurrence of a stimulus. And then we have cognition and problem solving. This is the highest form of learning that is available only to very, very smart animals, things like crows, chimps, dolphins, and of course us. Some other animals can do that too. And even without trial and error, animals like chimps can figure out solutions to problems. Here we have tool use. We have a chimp using a termite stick, kind of figuring out the right shape and size of the stick so we can stick that into a termite hole, pull out some tasty protein-rich termites. And that was not a result of trial and error. It's a result of just looking at the hole, looking at the sticks, and kind of figuring it out just by looking at it. Problem solving over here, we got the crows hanging from a string as a piece of meat. And other birds, other predator birds or birds that like meat, will try to fly at the meat and they'll miss it. However, the crow, just by looking at it, thinks, oh, I'm not going to be able to fly and grab that thing. So what they do is they pull it up a little bit at a time with their claw, and using their beak claw, are able to pull up that meat and get their food. And they're able to do that all without any help. And that's cognition and problem solving. Social behaviors are usually contests for resources, basically a pecking order of some kind. However, they're usually not to the death. So when two things animals are fighting each other, if it was to the death, even the winner might be mortally wounded. That's not going to provide a survival advantage. So there's not a whole lot of selection for anything other than maybe a ritual display of being an alpha male. A lot of times this is for mates or resources for these kind of social behaviors. Here we have a dominance hierarchy, a social ranking in a group, pecking order. Just means that there's one animal that's kind of above the other ones, called an alpha male. You have that in many social animals. Animals that are solitary typically don't have as strong a dominance hierarchy. All right, social behaviors. We have altruistic behaviors, or why would animals act nice to another animal? Well, it involves kin selection. If all the family members, your kin, have the same gene for the warning of making noise or stomping your foot or whatever it is, that allows you to warn your fellow family members of danger, then the gene survives, even though it does provide a slight disadvantage to you if you're the one calling out that warning, because that's going to attract attention to you. Now, this only works if it is a gene that's common in the population, and that's why it's called kin selection. If the other members don't share that gene and you're doing all the warning, you're just more likely to take yourself out of the gene pool because you're attracting attention and there's no gene to be selected for. Pheromones are chemical signals, especially in simpler animals, but also in some complicated animals, that are used to act as a stimulus. We have alarm pheromones. One fish detects danger, and they avoid the alarm pheromones, try to get away from it. Sex pheromones do the opposite, attract mates. Honeybees communicate by doing something called a waggle dance. You don't have to know anything specific about this, but they do have this little dance or an activity that shows the direction of a food source. And it's like an angle that will let the other bees know where to go. In auditory communication, bird songs are used for species identification. The thing that you need to know about bird and insect songs is that they're both involved with things like mating. However, bird songs are learned. It's an imprinting thing, but it's an instinct learning. This is during the younger years of a bird's life, so it's only during a very short window of that bird's development. Insect songs are developmentally fixed, they're genetically controlled, they're innate behavior. They don't change those songs no matter what. All right, this ends part one of your review for quarter one AP Biology.